The first method is for cell culture and use IFA to identify the virus. We inoculate the virus into the cells. As you can see here, the virus infects the cell become rounded like a tear-like cytopathic effect we call CPE. Comparing to the mock invasion, the cell is quite normal and quite different from the CPE induced by EV71. And how to do the immunofluorescence assay? Because there are so many different kinds of enteroviruses, and it's impossible to use every antibody to identify the enteroviruses. So in the beginning, we will use an antibody called pan-entero antibody. This antibody is quite special. It can recognize every kind of enteroviruses. So if the specimen contains enteroviruses, we will know if it's positive signal, we call it enteroviruses. After we got the positive signal, we will use the group-specific blend antibody for diagnosis, such as Coxsackie A or Coxsackie B or ECHO or numbered enteroviruses. Once we got a positive signal, then we will use the specific antibody for final confirmation. For example, EV71 specific antibody to know the patient got infected by EV71. So this is the flow chart, how we use IFA, immunofluorescence assay, for identification and typing.
After the specimens are transported to the laboratory, we will amplify the virus in the specimen for further identification. First step, cells were seeded at monolayer confluency before virus infection. Virus is taken out from the 80 degrees Celsius before zero refrigerator and thaw the virus via gentle agitation in 37 degrees Celsius water bath rapidly. After ice crystal melt, place the virus on ice. Cells are taken out from 37 degrees Celsius incubator and then moved to biological safety cabinet. Remove the cell culture medium from the culture dishes. Wash the cells twice or once with PBS. Mix the virus by pipetting. Add appropriate volume of virus to provide an optimal multiplicity of infection, short for MOI, to the monolayer of the cells, and then incubate at 37 degrees Celsius incubator for an hour. After an hour, remove the virus. Wash once with PBS, and then add appropriate medium containing 2% FBS to the culture dishes. Place the dishes to the 37 degrees Celsius incubator. After the appropriate incubation time period, infected cells were checked via microscopy to confirm the morphological changes for cytopathic effects, also called CPE. Because the genome of enteroviruses is an RNA, we will extract the viral RNA using trisol reagent. In biological safety cabinet, 0.75 milliliter of trisol LS reagent were mixed with per 0.25 milliliter of virus volume in microcentrifuge tube, and then homogenize the sample by gently pipetting several times. Incubate for 5 minutes. To avoid RNA's contamination, we recommend to wear mask and gloves. Wipe the laboratory bench, micropipette, and work surfaces with RNA ZAP RNase decontamination solution. Add 0.2 milliliter of chloroform to the sample in the fume hood cabinet. Mix sample by inverting the tube several times, and then incubate for 2 to 3 minutes at room temperature. We can observe the mixture separates into two phases initially. Centrifuge the sample for 10 minutes at maximum speed at 4 degrees Celsius. After centrifugation, the mixture separates into three phases, including a lower red phenylchloroform, interphase, and a colorless upper aqueous phase. Extract and transfer the upper aqueous phase to new microcentrifuge tube carefully. Avoid to contaminate with other phases. Add 0.5 milliliter of isopropanol to the aqueous phase, and then mix the sample by pipetting several times. Incubate for 10 minutes at room temperature. Centrifuge the samples for 10 minutes at maximum speed at 4 degrees Celsius. Aspirating the supernatant with micropipette, and avoid contacting with a white gel-like pellet at the bottom of the tube. prepare 75% ethanol and resuspend the pellet with 1 milliliter of 75% ethanol. Centrifuge samples at maximum speed for 10 minutes at 4 degrees Celsius. Aspirating the supernatant with micropipette. Air dry the RNA pellet for 5 to 10 minutes. Resuspend the pellet in 20 to 50 microliter of RNase-free water by gently pipetting up and down. Incubate in heat block set at 55 to 60 degrees Celsius for 10 to 15 minutes. Determine the RNA yield and quantity using the nanodrop TM spectrophotometer. Absorbance of 260 to 280 ratio of about 2.0 is generally accepted as pure for RNA. The 260 to 230 ratio for a pure nucleic acid is commonly in the range of 1.8 to 2.2.